Hello, good afternoon. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, part of the Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the webinar website for more details. I'm Martin Poole. I'm the local ambassador representative on the Society's Council of Trustees. Our invited speaker today is Dr. Sara Priego Moreno from the Salk Institute for Biological Studies. She's one of the Biochemical Society's 2020 Early Research Awards recipients, which are awarded to recognise the impact of research carried out in the molecular biosciences by early career scientists. Sara's research has focused on understanding the molecular mechanisms that govern the poorly characterized termination stage of eukaryotic DNA replication. Using egg extracts from the African clawed frog Xenopus laevis to run in vitro DNA replication reactions, Sara was able to characterize for the first time the biochemistry underlying one of the most important events taking place during replication termination, namely replisome disassembly upon replication fork convergence. The CMG complex is the eukaryotic replicative helicase that performs parental DNA unwinding during DNA replication and also functions as a central platform for the assembly of the replisome. It has become clear that assembly of the CMG helicase onto chromatin for the assembly of at the first stages of DNA replication is strictly regulated during the cell cycle through a well-characterized molecular mechanism in order to protect genome integrity. However, the mechanism of CMG disassembly during a DNA replication termination, as well as its importance for the maintenance of genome integrity, remains poorly understood. Today, she'll be presenting her award-winning research in regards to the mechanisms of CMG disassembly and their importance for the maintenance of genome integrity, as well as emerging questions and future directions. Questions will be asked at the end of the webinar, but you can send in your questions already during the talk. If you have a question, please type it into the question box as shown on the image on the screen at the moment. Hopefully you can see that. Okay, so without further ado, I'll hand over to Sara. I'm very much looking forward to hearing her presentation.
Okay, perfect. Thank you. Okay, well, thank you, Martin, for your introduction. And also I want to say a big thank you to the Biochemical Society for choosing me as a recipient of one of their early career research awards. And today I'm going to tell you about the work that I did during my first year as a researcher in the laboratory of Aga Gambus at the University of Birmingham. And during these years, I studied the mechanism by which the replicative helicase is disassembled during DNA replication termination. So that's what we are going to talk about today. Okay, so DNA replication is a very fundamental process. Every cell before cell division needs to duplicate the DNA in order to give an exact copy of genetic material to each daughter cell. And DNA replication, normally, DNA replication is a very accurate process and it has a very high fidelity. And this is very important because we now know that most of the genomic instabilities that are found in cancer are due to errors during DNA replication. So it's crucial that we understand in detail how the process of DNA replication works. And in eukaryotic cells, DNA replication happens during the S phase of the cell cycle, and replication starts from origins of replication, which are locations within the genome where replication starts. And origin activation well, there are multiple origins in eukaryotic cells, and activation of these origins doesn't happen simultaneously. Instead, they follow a specific replication timing program, which means that some of these origins are activated earlier during the S phase, some of them are activated later. And eventually, this leads to the replication of the entire chromosomes that are then ready to be pulled to each daughter cell during chromosome segregation. And the regulation of the activation of these origins is very, very well regulated during the cell cycle in order to maintain genome integrity. And the regulation of origin activation is by a two-step mechanism separated into two different cell cycle phases. The first stage of this mechanism is known as origin licensing and takes place during end of mitosis and G1 phase. And it consists on the loading of the MCM227 complex onto DNA. Now, this MCM227 complex is the core of the replicative helicase that unwinds replication, that unwinds DNA during replication. However, in this state, this helicase is inactive, and it can only be activated upon S-phase entry, because only in this phase of the cell cycle, the recruitment of these additional regulatory subunits, CDC45 and genes, takes place. And all of these proteins together form the CMG complex, which is the active helicase that can promote DNA unwinding. So once the helicase have been activated, they now have the ability to translocate through single-stranded DNA and unwind the double helix. And this allows for the exposure of single-stranded DNA and DNA synthesis by the DNA polymerases. And eventually, what happens is that a CMG complex coming from one origin will encounter another CMG complex coming from the neighboring origin. And at this stage, termination of DNA replication takes place. Now, in contrast to the early stages of DNA replication, the termination of DNA replication remained very poorly understood. However, in 2014, we were able to characterize one of the main steps that occurs during this process, which is the disassembly of the CMG helicase upon convergence of replication forks. We showed that this mechanism was strictly regulated by ubiquitination, and we showed this together with the Labib lab, who also studied this in yeast. And this is what we will be talking for the majority, or the rest of my talk. And I just want to mention also that shortly after our publication, the Walter lab came up with other aspects of the mechanism of termination DNA replication, which generated the first model of how termination happens. And they showed that during termination, the helicases actually finished to unwind the DNA between them, and they actually pass one another in the termination site. And then they showed that disassembly of the replicative helicase was the last stage of replication. So anyway, I will be telling you what we discovered about this mechanism of CMG disassembly. And in the Gambus lab, to study DNA replication, we used Xenopus egg extract. So Xenopus egg extract is an extract that we made from eggs, the unfertilized eggs that we collect from Xenopus laevis, which is a frog that you can see there in the picture. And basically, to make these extracts, what we do is to collect the eggs. We process them by centrifugation steps and other things. and once and then we can extract the cytoplasmic fraction of this X, and this is really the extract that we used, and it is very good for the study of DNA replication because it contains a very rich stock on cell cycle factors and other proteins that are required for the replication of DNA. So basically, if you take this extract and you add Xenopus sperm DNA to it, this can serve as a template, and DNA replication will happen in the extract, and it will recapitulate all of the steps that happens during DNA replication in cells. So it's a very efficient system. So what we first found out is that ubiquitination was essential for the disassembly of the CMG complex during termination. So ubiquitination, it's a post-translational modification that consists on the attachment of a small protein called ubiquitin to a substrate protein, and it does so by attaching covalently to a lysine residue within the substrate protein. And because ubiquitin itself has 7 lysines within its sequence, these lysines can serve as acceptor lysines for the attachment of further ubiquitins. And in this way, ubiquitin can form chains. And depending on which lysine residue within ubiquitin is used for the formation of these chains can have different conformations and different roles in cell biology. So in order to see what was the importance of ubiquitination in DNA replication, what we did was to add recombinant ubiquitin, either wild type or a mutant of ubiquitin that had all the lysines mutated, so it couldn't form chains. So that was our UBNOC mutant. So we did that, and we looked at the ability of the extract to replicate DNA by measuring the incorporation of radioactive nucleotides into nascent DNA. And we could see that inhibition of ubiquitin chains formation in the extract didn't affect the ability of the extract to replicate DNA. However, surprisingly, what we observed is that when we inhibited polyubiquitination, we could observe that components of the CMG complex were associated with chromatin for an extended period of time in comparison to the control, to wild-type ubiquitin. So here what we are looking at is at chromatin or DNA samples isolated from the replication reactions through a replication time course, different time points. And we did a lot of work to figure out why we could be seeing this phenotype, and we showed that this wasn't due to a problem on replication fork stalling, or it wasn't due to increased origin firing or re-replication. Instead, we showed that the reason why we saw the stabilization of the CMG complex on chromatin is because the CMG disassembly mechanism was impaired. And we also showed other things. We showed that actually this polyubiquitination was ubiquitination through lysine 48 ubiquitin chains. And we also showed that the MCN7 subunit of the CMG complex was the only subunit that we found ubiquitinated. And then I didn't mention this before, but ubiquitin chains linked through lysine 48 are very well known for their role in targeting proteins for proteasomal degradation. So we thought that maybe disassembly of the helicase was driven by proteasomal degradation of the CMG complex on chromatin. However, when we inhibited the proteasome, the CMG disassembly process still happened. So it was something else. So in order to see what was driving a CMG disassembly upon ubiquitination, what we did was to study which were the proteins associated with the terminated with the CMG complex during S phase. And what we found is that it associated with the protein P97. We did this by immunoprecipitating one of the components of the active helicase from chromatin, actually. And we could see how when we immunoprecipitate MCM3, we have other components of the CMG helicase, such as MCM7, CDC45, and P97. And this was very interesting because what P97 does is P97 is a AAA ATPase that it associates with a series of cofactors. And these cofactors help P97 to be recruited to ubiquitinated substrates. And by using its ATPase activity, it actually drives the translocation of the ubiquitin chain through the central pore of P97. And by doing that, it promotes the unfolding of the substrate protein, and it segregates the protein from the other proteins that it might be forming a complex, or from cellular structures such as chromatin or membranes. And here you can see a crystal structure with P97 in a complex with a ubiquitinated substrate. And you can see how the ubiquitin chain goes through the central channel of P97. So anyway, P97 was a very good candidate for disassembling the ubiquitinated helicase. And in order to test whether P97 was actually involved in this process, what I did was to purify from E. coli either a recombinant P97 wild type or P97 mutant, a mutant version of the protein that didn't have, couldn't hydrolyze ATP. And what we observe is that upon addition of these proteins to our replication reactions, DNA replication proceeded well in the extract, as we saw before when we inhibited polyubiquitination. However, we recapitulated the CMG disassembly defect that we observed upon inhibition of ubiquitination. So yeah, and we could also observe an accumulation of ubiquitinated forms of MCM7 on chromatin. And we saw this by adding his tag ubiquitin to our replication reactions, either in the presence or in the absence of P97 mutant. And then what we did was to pull down the ubiquitinated proteins from chromatin using magnetic beads. And we could see that when the activity of P97 was impaired, we could see huge accumulation of these ubiquitinated forms of MCM7 on chromatin. So all of these results, we published them, and this is the model that we came up with the first, showing the first elements of the disassembly of the CMG complex during termination. And basically, as I showed you before, we showed that during termination, the CMG complex was ubiquitinated with lysine 48 linked ubiquitin chains on the MCM7 subunit. and this led to the disassembly of the complex by P97. And we published this in 2014 in back-to-back with work from the laevis lab, who also found the same mechanism to happen in budding yeast. So this is actually a mechanism that is very well conserved throughout evolution. And then the next questions that we asked was, well, what it is the ubiquitin ligase that is driving this ubiquitination of MCM7? And what we knew is that the ubiquitin ligase responsible for CMG disassembly and MCM7 ubiquitination in budding yeast from work from the laevis lab was the ubiquitin ligase STF-dia2. Now, STF-dia2 is a Kalin ring type ubiquitin ligase, and these type of ligases are multi-subunit protein complexes that are built around a Kalin scaffold protein. In the case of STF-dia2, the scaffold protein is CDC53, and the substrate receptor subunit is dia2. However, in higher eukaryotes, dia2 doesn't have a direct homologue. So we didn't have a direct candidate that we could test in Xenopus egg extract. What we knew, however, is that the activity of Kalin ligases was essential for the disassembly of the replicative helicase and for MCM7. Because when we used a small molecule inhibitor against the activity of this family of ubiquitin ligases, we abrogated both MCM7 ubiquitination and CMG disassembly. However, in higher eukaryotes, there are at least 6 different Kalin subunits, and each of them associate with different pools of substrate receptors. So really, there were many different ubiquitin ligases that could be driving this mechanism. So in order to identify the ubiquitin ligase that was driving CMG disassembly during termination, what we did was to study which were the proteins that were associated with the CMG complex during termination. And to do that, what we did was to replicate DNA in the presence of a P97 inhibitor. So after running replication in our extract and DNA synthesis was completed, what we had was a lot of CMG complexes accumulated on chromatin that were ubiquitinated, and all of the other interacting partners that are around the CMG. So we isolated the chromatin and we immunoprecipitated the CMG helicase, and then we studied which proteins were associated with it through mass spectrometry. And here is just a short list of what we found associated with it, but you can see how we obtained all of the components of the CMG complex, and also all of the components of this ubiquitin ligase, in which Kalin2 is the scaffold protein, and LRR1 it was the substrate receptor. So we also confirmed these interactions in a Western blot. Here you can see that when we immunoprecipitate NCM3, we don't only immunoprecipitate the components of the active helicase. You can see here the ubiquitinated forms of MCM7, but we also immunoprecipitate Kalin2. And we did this the other way around by immunoprecipitating LRR1, the substrate receptor, and then we also co-immunoprecipitated with it the components of the CMG complex. So then the thing is that we saw Kalin2 LRR1 associated with the CMG complex during termination, but we wanted to confirm that this was the ubiquitin ligase that was driving ubiquitination of MCM7. And to do that, we wanted to remove the activity of Kalin2-LRR1 in Xenopus egg extract. And to do that in Xenopus egg extract, we used immunodepletion. And the way this works is by coupling antibodies against the particular protein that you want to immunodeplete, in this case Kalin2, to magnetic beads. And what you do is to incubate these beads with the Xenopus extract, and you will recruit all of the Kalin2 proteins, and then using a magnet you can recover these beads and remove all of the proteins from Kalin2 protein from the extract. So here is, so we do a couple of rounds of this incubation of beads with extract. And here you can see in the first incubation, you can see how these are the beads recovered after immunodepletion, and you can see how Kalin2 and LRR1 are bound to the beads. And this is how the extract looks like. You can see how after depletion in the mock extract, in the control extract, you have Kalin2 LRR1, but not in the Kalin2 depleted extract. So we could observe that Kalin2 depleted extract were totally fine, were fine in the replication of the in the DNA replication. However, we could recapitulate the defects on CMG disassembly that we observed upon inhibition of ubiquitination or P97. And we could also see that the ubiquitinated forms of MCM7 were restricted in this extract. However, when you do immunodepletion, when you do these types of immunodepletion, you need to be very careful interpreting your results, because the phenotypes that you see could be due to the co-immunodepletion of other proteins by accident. So in order to confirm that Kalin2-LRR1 was indeed required for ubiquitination of MCM7 and CMG disassembly, what we did was to try to rescue these phenotypes that we observed by adding back a recombinant Kalin2-LRR1 complex. So here, what you can see is that when we replicate DNA in an extract for 2 hours, DNA replication has been completed. And in the control extract, if we look at the CMG complex on chromatin, we can see that the CMG complex have been efficiently removed. However, this wasn't the case in Kalin2 depleted extract, as we saw previously. Then, when we add back either recombinant, either wild type recombinant Kalin2 or mutant Kalin2, we could see that these phenotypes were not recovered. However, when we added also a recombinant LRR1, we could see that this phenotype was rescued in the presence of the wild type Kalin2, but not in a mutant of Kalin2 that cannot promote ubiquitination. So not only Kalin2-LRR1 was required for CMG ubiquitination, but actually the activity of Kalin2-LRR1. So it was the ubiquitin ligase driving MCM7 ubiquitination and CMG disassembly during termination. And what we also studied was the regulation of the recruitment of the ubiquitin ligase to the CMG complex, because we didn't know if the ligase was associated with the CMG complex always or only during termination. So to answer this question, we look at the recruitment of Kalin2-LRR1 to the CMG complex upon different treatments in the extract. And we could see that, well, in control extracts, we could see sometimes the presence of Kalin2-LRR1 on chromatin at the same time as we observe other components of the CMG complex. And we could see calin-2-LRR1 stabilized on chromatin when we stabilized the CMG complex by inhibiting its disassembly with a p97 inhibitor. But however, what we could observe is that when we stabilized the CMG complex on chromatin by preventing convergence of replication forks, we didn't see the ubiquitin ligase on chromatin. And what this means is that the ubiquitin ligase calin-2-LRR1 is only recruited to the CMG complex when replication forks are allowed to converge and terminate. So it's a very regulated process. And again, these treatments didn't affect the replication efficiency of the extract. So again, we published this in 2017, and this is the model that we came up with. So we showed you that upon replication termination, the ubiquitin ligase calin-2-LRR1 is specifically recruited, and this promotes the ubiquitination of MCN7 and the disassembly by p97. And we published this in 2017 in collaboration with the lab of Karim Labib, who also found the same elements of this mechanism in C. elegans. So this was very exciting. It was very nice, and yeah, and this was also, similar results were also published by the Walter lab, also in Xenopus laevis extracts. And an observation that that the lab did in early embryos of C. elegans is that when they depleted calin-2-LRR1 in the embryo, what they could see is that the CMG complex was stabilized on chromatin during S phase. However, upon mitotic entry, the CMG complex was rapidly disassembled in a reaction that was dependent on p97 and also other factors such as ULP4 and UBXN3. So it seemed like in mitosis, there was like a backup mechanism to allow the replisomes in the case that in S phase, it didn't happen. However, this pathway of CMG disassembly during mitosis at this stage was poorly characterized. So we were very interested in understanding how this worked and what were the elements of this pathway. And to do that, we decided to study this pathway in our extract, in Xenopus egg extract. And the approach that we followed for this was to replicate DNA in an extract that was supplemented with an inhibitor of the activity of calins. So in this extract, if we allow replication to be completed, we have replicated DNA with CMG complexes trapped there that were not fully ubiquitinated because of absence of calin activity. And what we do is to push mitotic entry at the end of DNA replication by addition of cyclins, recombinant cyclins. And then we studied what happened with the CMG's complex on chromatin. And what we could observe is that while in a control situation where we just had a buffer control, the CMG helicase remained associated with the chromatin forever, for a very long time, you can see here. However, when we pushed this extract into mitosis, we could see that the CMG complex was now disassembled. So we could also see this mitotic pathway of CMG disassembly in Xenopus egg extract. And what we could also show is that this mitotic pathway will require the activity of P97, because when we inhibited P97, we abrogated the disassembly of the CMG complex. And what we could also observe is that now MCM7 presented these higher shifts that we thought it looked like ubiquitination. A ubiquitination that happened specifically in mitosis. So in order to test whether these shifts were actually ubiquitination and whether ubiquitination was required for the disassembly of the CMG complex also in mitosis, we again used the same ubiquitin mutant that I mentioned at the beginning of the talk. And we could observe that upon addition of this mutant to the extract, the components of the CMG complex was not as disassembled as it was in the presence of the wild-type ubiquitin. And also, we couldn't observe anymore these very long shifts of MCM7 ubiquitination in the extract. So it seemed that ubiquitination, this shows that ubiquitination was actually required for mitotic CMG disassembly and this extra shift of MCM7 ubiquitination. And we also showed that this ubiquitination wasn't linked through lysine 48 as it happened in S phase. It was a different type of ubiquitination. It was ubiquitination linked through lysine 6 and lysine 63. So this also means that different ubiquitin ligase was driving the disassembly of the CMG complex, specifically in mitosis. And we followed a similar approach as I showed you before for the identification of this ubiquitin ligase. So what we did basically was similarly as for the identification of calyptular R1, we replicated DNA in the presence of a P97 inhibitor. We allowed replication to be finished. And also after that, we drove mitotic entry by addition of recombinant cyclins. And we isolated chromatin with the accumulated helicases, and we isolated this helicase by immunoprecipitation and studied the proteins associated with it. And we could find this time that the CMG complex associated with the ubiquitin ligase trait. Here you can see the confirmation of this interaction by Western blots. And we also did all the experiments to show that actually the activity of trait ubiquitin ligase was required for mitotic CMG disassembly and MCM7 ubiquitination during mitosis. So this led us to propose this model in which when termination happens in the absence of calin activity, calin-2 activity, the replisomes, the CMG complexes are allowed to terminate. However, CMGs remain on chromatin, but when mitotic entry happens, the ubiquitin ligase trait becomes activated, and it promotes the ubiquitination of MCM7 with lysine linked through lysine 6 and lysine 63, and this leads to P97 dependent CMG disassembly. And not only that, but our lab also showed that not only the terminated CMGs were disassembled by TRAPE and P97, but also CMGs that were stalled during S phase and didn't manage to complete replication during S phase. So when this happened, the CMGs remained on chromatin through S phase, but in mitosis, they were also unloaded by this trait P97 mechanism. And the Labib and the Walter Lab, their work also showed, suggested that this mechanism was actually like this mechanism could be actually a way to process under-replicated DNA during mitosis, by allowing cleavage of the DNA strands that are otherwise protected by CMG and promoting repair of this repair of this unreplicated DNA during mitosis with mitotic DNA synthesis. And so this was all published recently in 2019 by our lab. Then the Walter Lab showed this breakage and repair mechanism, and the Labib lab and the Labib lab showed that this that trait was required for mitotic DNA synthesis. And now you might wonder why it is important to have a mechanism to specifically unload the helicase during termination. And this is actually due to the fact that the helicase can only be loaded during the G1 phase of the cell cycle. And once S phase entry happens, the CMG complex can be activated, but loading of the helicase can't happen anymore. So, as you can imagine, the presence of the CMG complex on chromatin during S phase needs to be very stable, because if we lose it, we cannot just reload it during this phase. So it needs to be very protected on chromatin until it has done its job and DNA replication has finished. Then the disassembly can happen. So it's a way to make sure that CMGs are protected on chromatin until they are not needed there anymore. So it is very important. And what we still don't fully understand is what are the consequences of deregulating this process what are the consequences for genome integrity of deregulating CMG disassembly. But you can imagine that many things can go wrong. For example, if we don't disassemble efficiently CMGs upon termination, and cells go through the cell cycle and move on to the next cell cycles, these cells would have plenty of CMGs on chromatin that could interfere with other DNA metabolic processes such as DNA replication and transcription, and this could be very problematic. And also, you can imagine that if the trait mechanism for unloading stalled CMGs during mitosis was impaired, the processing of under-replicated DNA during mitosis would also be impaired, and this could also have very bad consequences during chromosome segregation, because it could lead to random chromosome breakage and to problems in segregation that could lead to different numbers of chromosomes taken by each daughter cell during cell division. So many things could happen. And we actually know that the regulation of the ubiquitin ligases involved in this process is associated with genomic instabilities. However, these ligases also have other substrate and other roles, and we cannot exclude that the genomic instabilities that we observe when these ligases are deregulated are due to a defect in CMG disassembly. So in order to understand what is the impact of the CMG disassembly mechanisms, we need to come up in the future with approaches that target this process more specifically. And this is a challenge for the future. With this, I just want to say a big thank you to everyone in the Gambus Lab, especially, to everyone in the Gambus Lab, but especially to everyone that contributed during this, with this research, because this was really teamwork, and it couldn't have happened without the contribution of everyone. And I also want to give a big thanks to my current lab, the lab where I'm doing my current postdoc, the Carl Sader Lab. We are in San Diego at the Salk Institute, and we are also having a lot of fun here. And I am here studying, apart from other aspects of telomere biology, I am interested in studying replication mechanisms at telomeres. So yeah, I thank you to all of the people that I work with. Science is great, and I'm having a great time. So with this, I'm ready to take questions.
Okay,
Thank you to EMBO and Human Frontiers for funding my current research.
Okay, thanks, Sara, for a great presentation. We've only had a few questions so far, so if you would like to ask Sara a question, please do free to type those into the question box. So the first question that I've got for you, Sara, is this: what is the signal that activates the ubiquitin ligation? Is the SCF complex brought to the termination site by replicative enzymes?
So, so this is actually something that we still don't understand. So it could be possible that upon termination of replication forks, because it seemed like from the model proposed by the Walter Lab, the CMG complex translocates through single-stranded DNA, but upon termination, it seems like it translocates to double-stranded DNA. So this could generate a conformational change, or other things that happen during termination could generate a conformational change in the CMG complex that exposed a motif that expose a motif or something on MCM7 or somewhere else in the CMG, and this allows recruitment of the ubiquitin ligase to the CMG. But yeah, we still don't know that for sure, because we haven't been able to map that motif and mutated
Thanks, Sara. We've not had any more questions. I had a question for you. So I know P97s have a range of different cofactors. Do you know if there's any specific cofactor that's actually is required for the role in the disassembly here?
Yes, actually. So actually, the Labib Lab has shown that UFD1 and NPL4 are required for this for the recruitment of the P97 protein to the ubiquitinated CMG during termination in budding yeast, where there is only one pathway of CMG disassembly depending on Calin2 on SDF dia2. And we have also seen these cofactors associated with P97 and the CMG helicase. However, what we don't know is that maybe in mitosis, there are other cofactors of P97 that are required to recognize the different ubiquitin chains built on this stage of the cell cycle on MCM7. But we don't know that. So the Labib Lab showed that in C. elegans, UBXM3, that is the homologue of FAP1 in humans, he showed that this protein was required for mitotic CMG disassembly, but we don't have any evidence whether it plays a role as a P97 cofactor. That's something that we also would like to understand in the future.
Okay, sure. So in your assay where you added back the recombinancy P97, did you need to add the cofactors with that, or was that just with P97 acting alone?
It was only P97 alone, but because these cofactors are present in the extract already, they could work with recombinant P97.
Okay, great. I've got another question from one of our attendees. So the cell cycle is governed with CDKs. Did you happen to study these and their role in this process?
No, I didn't. We just, I mean, the only thing I did with CDKs is not directly with CDKs, is when we induce mitosis by addition of recombinant cycling, but that is what this recombinant cycling does, will be to associate with actually CDKs that are present in the extract in order to promote entry into mitosis. But I didn't directly do any work with CDKs.
Okay. And then there was another question. I was wondering how culling LRR1 protein ubiquitinating CMGs only at termination. Is culling LRR1 protein expressed only at the time of termination, or is its binding or activity regulated?
Exactly. So that's exactly what we've been showing, that so culling 2 LRR1 is present there, but it's only recruited specifically to the terminated CMG complex. So it's the recruitment that is highly regulated.
Okay, and a follow-up part of that question: does the culling LRR1 have any DNA binding activity itself?
I don't think so.
Okay.
No, not that I know. It has the LRR leucine-rich repeats within LRR1, which is the motif that is thought to be the motif that regulates substrate recognition.
Okay. Another question. So, do all MCMs fire? Is there a subset that remain dormant? And if so, how are dormant MCMs removed?
So no. So actually, yeah, during G1, many a lot of MCMs are loaded onto origins, and there is actually an excess of MCMs that are loaded, and this is actually due to the problem that MCMs cannot be loaded during S phase. So you always want to have some backup MCMs on chromatin in case that some helicases become stuck, stalled. These MCMs can fire and rescue replication. I don't think we still understand the mechanism by which these dormant MCMs are removed from chromatin. I think they might be, so MCMs can slide on double-stranded DNA, and they can be pushed by the approaching CMG complex, and we still don't know how. I think we still don't know how these are removed.
Okay, and then next question. So you mentioned that ubiquitination of MCM7 in mitosis is mostly not via lysine 48, which is the CDC48's favorite chain type. If not, does that mean that CDC48 maybe needs a different adapter to deal with that situation during mitosis?
It could be perfectly, yeah, totally.
Okay, we've probably got time for just one or two quick. So how close to the terminus do CMGs need to get? Is it proximity based or based on the recruitment of other termination proteins?
Okay. Yeah. So actually, they move. So is the N terminus that is at the at the head of the so it's N terminus to N terminus, I think. And so actually they need to pass one another during termination, and the model right now is that they are the ligase might be recruited after they have passed one another during termination.
Okay, and then a final question: is the ubiquitination also involved in the assembly of CMG or just in a disassembly? And does CMG also undergo any other post-translational modifications?
So during the assembly, I don't think ubiquitination, no, I don't think ubiquitination plays a role. But in fact, it is phosphorylated. Several subunits of the MCM complex are phosphorylated. Yeah.
Okay. Thanks, Sarah, and thanks everybody for all of your questions. So just before we finish, I'd just like to stress that the Biochemical Society welcomes suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. So if you do have an idea for a webinar, please submit a proposal for an upcoming webinar via the website. I'd also like to trail our next webinar, which will be on Wednesday, the 29th of July at 2 o'clock, where Professor Dame Kay Davies from the MD UK Oxford Neuromuscular Centre, who's the 2020 recipient of the Biochemical Society Centenary Award, will present her lecture on the current status of Duchenne muscular dystrophy therapy, with a focus on those genetic strategies which have been taken forward to the clinic. And you can register, find more information about the webinars, also propose your webinar at www.biochemistry.org/webinars. So I just want to thank Sara again and everybody for listening and for posing your questions. So thanks everyone. That's the end of our webinar today.
Okay.